Okay, everyone. Hello and welcome to today's webinar, FDA GCP Inspection Trends Identified Using AI, presented by Redica Systems and Jerry Chapman, Senior GMP Quality Expert. Today's presentation was recently recorded. If you have any questions, please use the chat feature and we will have a live Q&A section at the end with Jerry and a special guest. After we're done, I'll send out a copy of the slides as well as answers to all the questions we receive and a recording of the presentation. Before we begin, I want to quickly introduce ourselves. My name is Taylor Minks and I am the marketing lead here at Redica Systems. Redica Systems is a technology company using data, analytics, and expertise to deliver meaningful insights to quality and safety professionals around the world. By applying artificial intelligence to large and disparate government data sets, Redica Systems empowers its customers to improve compliance, increase product quality, and build a more efficient organization. Founded in 2010, Redica Systems serves over 200 customers in the pharma, medical device, and food industries, including 19 of the top 20 pharma companies and 9 of the top 10 medical device companies. Today's presenter, Jerry Chapman, is a senior GMP quality expert here at Redica Systems. He writes the Redica Systems column, Conference Spotlight, and is working with our data engineers to build machine learning models for analysis of agency enforcement documents. He brings 40 years of experience in the pharma industry, including 31 years at Eli Lilly in technical and leadership positions, and research, product development, manufacturing, plant quality, site quality, corporate quality, and quality systems. As I mentioned, we will have a special guest for our live Q&A, who is Fran Lambetecchio. She is a GVP and GCP expert. She has held leadership roles as Senior VP and Senior Director in multiple biotech firms and is a subject matter expert in the investigations of fraud, misconduct, breach with clinical systems, and used for conduct of clinical trials. As a reminder, this is a pre-recorded webinar, but we will have a live Q&A section at the end. And now I will start today's webinar.
Hi, this is Jerry Chapman, Senior GMP Quality Expert at Redica Systems. And today I'd like to talk to you about evaluating clinical study deficiencies found during inspection and some of the artificial intelligence tools we use to do that. So to begin with, a little slide about my company, which I'm not really going to go into. You can look at that at your leisure. So I'd like to start with the importance and challenges of human clinical studies, and then kind of look at the overall inspection landscape from a macro level. And then the data sources that we use for inspection information, how we evaluate them. And based on our evaluation, what have we learned from warning letters to clinical investigators and also from 483s. So to begin with, this audience knows better than I do the importance of human clinical studies to individual and public health. For the purposes of this presentation, just a reminder of the importance of the data generated and how it is a key part of FDA approval of the drug or device. And there are a number of challenges, again, which you know all too well. I will point out that the ones on this slide in bold are challenges that Redica Systems can help you meet. Another challenge is keeping up with how the agency is conducting inspections, and agencies does publish guides that are helpful to us. And there was one that was published with little fanfare really back in September, this FDA BIMO inspection guide. If you haven't seen it, I would encourage you to look for it and find it on FDA's website. It has an increased emphasis, among other things, on selecting and monitoring clinical investigators, including understanding their compliance history, and selecting and making changes in the principal investigator. Both of which are some things that we can help you with. And I'll point out here that at the bottom of a number of these slides along the way, you'll see sentences that are in red, bold, and those are key points of that particular slide. So let's look at some deficiency statistics. Now, these are from FDA, and this is simply the numbers of these inspections over the last 6 years. And we're looking not at the REMS and GLP and bioequivalence inspections, but at the clinical study inspections of clinical investigators, IRBs, and sponsors, CRO sponsor investigators. And when you look at that data set, what you see is that clinical investigator inspections comprise about 75% of those. And when you add the IRB to that, the two together are about 87%. So those are the two categories that we chose to tackle to begin with. Now, if you look at those inspections, those numbers, and how they are apportioned between 483s and warning letters, you can see about a third of BIMO inspections resulted in 483s, and fewer than 1 in 100 resulted in a warning letter. So this will be important as we go through the data sets and the size of those data sets, right? The size of the warning letter data set is not very big. There are a lot more 483s. And this should be familiar to all of you. The number one inspection citation or observation rather in 483s is always 21 CFR 312.60, protocol compliance, every year. And the question is, what is the detail behind that? We really need the detail behind what are the aspects of protocol compliance that we need to focus our attention on that are being found deficient. So I'm going to look at our data sources and some of the tools we use. I'm not going to talk a lot about this, but I will say that all of our data is from public sources, but they're not easy to find and they don't all talk to each other. And there's a lot of work that we need to be able to prepare them and be able to use them, which we'll talk about to some extent. We have over 50,000 483s and 483 responses and EIRs together. And we have a team of dozens of data scientists and engineers, as well as subject matter experts in the various GXP areas to build our models and interpret this data. We can tag sites so we know based on an address what takes place at a particular location. And then there are a number of ways you can see in the right-hand column that we use this data. And we're going to be talking about a few of those. So key takeaway here is that Redica Systems has more publicly available documents in these areas than any non-governmental entity. So we have the data. What do we do with it? We have built expert models, and these are models, we call them expert models because they allow a computer algorithm to analyze documents the way that an expert would. They're created using machine learning and natural language processing tools. We have to prepare the documents for examination, so you can't just take these raw documents. There are ways that they need to be prepared using natural language processing tools that can help a computer be able to understand and interpret them, because as you know, a computer can't read documents and make sense of them the way that a human can, but they can analyze them if they've been properly prepared using these tools. So to begin with, a couple of the things that we do, I'll talk about a few of these tools. One is making picture file documents searchable. So if you have a document and it's scanned, it's just a picture. As a human, you can read it, but you can't do like a Control F and find terms in it. You can't search through it with a computer. The computer can't read it, the machine can't read it. So we need for them to be machine readable for analyses. So we clean the observation text. We use optical character recognition and proofing and retyping to put these into a form that we can analyze. We also do something called parsing. Text parsing is extremely important. If you look at something like a warning letter, it has a number of different parts, and each of those parts has a function, and each of those parts has certain kinds of language and certain properties in it. And if the algorithm's searching in the wrong place, you're going to get a lot of false positives, right? So these need to be able to be parsed into logical sections to allow appropriate analysis. So here's an example, what we call the deficiency title. You will recognize as being from a warning letter. It's usually bold and numbered and has boilerplate language. This is the most familiar one, right? You failed to ensure that the investigation was conducted according to the investigational plan, 312.60. Then it generally says specifically and gets into examples that were found during that inspection that support this citation. So it's really the specifically or the for example text that contains the content that allows us to drill down and get more granular. So let me give you an idea of what that looks like. Here are a few examples under 312.60 that we have found. So, for example, subjects were enrolled despite meeting exclusion criteria, didn't follow protocol-specific recommended guidelines, had material that was stored in a refrigerator that had a temperature excursion, failed to perform certain procedures that you were supposed to perform, issues with subject eligibility, issues with dosing and labs. Those are just a few of the examples. And I'd like to take this time to point out that it's can be a common misconception in the industry that if you get a 483 or a warning letter, you can determine how serious it is by how many observations are in it. And that is simply not true. You can't say, well, I had 2 observations and this guy had 4, and so his is twice as bad. It's not the case, right? It depends on the content of those observations. So it's something that's important to keep in mind. So we build the models to do these analysis. We create things called n-grams, and you saw some of those in the previous slide highlighted in green, that have particular meaning that we look for, that the algorithm looks for. We use Turbo EIR 43 text and some other things. So we have models that experts have built for human drugs and for medical devices. Those have been built, those have been tested, deployed. We have those, our clients use those and find them very useful for really getting at what the specific observations are and what the specific issues are, 2 and 3 and 4 levels down from that top observation. So the models are similar. They differ in a few ways because they're different GXPs. And today we're going to talk about GCP models. And to begin with, let's just go through briefly how those are organized. So you can see here in red, those are the top level or level 1, top high level buckets, if you will, of observations, in the red for IRB and in the green for clinical investigators. And then there's one category that is shared between them, data integrity. And we're going to talk about that today. Actually today, what we're going to focus on is the clinical investigators. You can see there are 2 really broad buckets. Responsibilities, sponsors, investigators, and informed consent. So we're going to take a look at what our models have found. To begin with, we're going to look at warning letters to clinical investigators. So the population we're looking at is 25 clinical investigator warning letters that have been issued in the last 6 years. And here's some of the results. So to begin with, and this is not surprising, the biggest bucket, right, or level 1, is responsibilities of sponsors and investigators. 22 of the 25 warning letters contain that as a citation. Informed consent in 4, and data integrity only in 2. So, as is perennially reported, responsibilities of investigators, right, is the highest level finding. But as we look at our models and apply our models to these 25 warning letters, what does that look like? Well, this is what our model gives us. So we have 63% fall into conducted according to the investigational plan, although there are 3 other subcategories that also fall under 312.60. So we're around 70%. that fall under that category. And then you can see the others as well, some of the other big slices of the pie. Remember, this is in two large buckets. So this is the big bucket, responsibilities of investigators, and how that's broken down. So the big one is 312.60, protocol compliance. What do the regulations say? Well, as you're aware, the regulations say conducted according to signed investigator statement, investigational plan, applicable regulations. I have that highlighted in blue. That's always number one in the observations and 483s, right? And then a little bit on informed consent, which is generally down around number 8 or number 9. So what we have been working toward is finding out what are the issues, the aspects of protocol compliance that are being found deficient. And here's what our model has come up with, right? These are the areas, this is an actual useful breakdown of the 312.60 citations based on our model using the tools that I've talked with you about today. And here are the areas. So the biggest one is having to do with various aspects of dosing, right? And you can imagine there are a variety of different ways where dosing could be an issue. Followed by inclusion and exclusion criteria, issues with doing that appropriately. And then we have this personal attention, which is really oversight by the principal investigator. And as you know, there's a lot of things we're supposed to have oversight of, and they're cited in a number of the warning letters for not doing that the way FDA thought they should. Issues with the lab, and those are things like labs being read when they're supposed to, conducted on time, various types of assessments that need to be done that haven't been done appropriately, and then adverse event reporting. So these are the big areas that our model has found that come under 312.60 protocol compliance. So those were the warning letters. Let's look at 483s. Okay. 483 data set is much larger. We have over 1,200 483s that were issued to clinical investigators over the last 20 years. If you put this into a Word document, it's over 6,700 pages and over 2.7 million words, right? So it's a sizable data set. And as we turn our model to this data set, what do we see? The model found that nearly 1,000 out of those 1,200 had issues that fall under our broad umbrella of responsibilities of the investigator. However, over half of them had issues with data integrity. That may be something that you haven't really thought about a lot. And I'm going to explain what that is and why it's important, right? And then, over 10% of them had issues with informed consent. So let's break this down as we did with the warning letters, responsibilities of the investigator. So we have the same, the pie sliced into the same sections, and the percentages are a little different. And you can see that I've listed there on the pie chart how those compare to the warning letters, keeping in mind that there are only 25 warning letters. So it's a much smaller data set, whereas we have 1,200, right, 483s. Again, it would be helpful to know what specific protocol violations are being found and noted, and what that looks like in the 483s. And so here's what our model has given us. Same major issues, and pretty similar in how many, the percentage of those issues, except for the personal attention or oversight given by the principal investigator. And we could go into that if you'd like. I'd be glad to talk about that. I'll have my contact information or during the Q&A, why I think that discrepancy. But this is really, these are the areas that we recommend you focus attention on in your operations. right? So if you're looking at the biggest issue FDA is finding on inspection and how that breaks down, these are the areas. So we would recommend focusing your attention in your operations on these particular areas to ensure that there aren't any issues or gaps. Now, let's go back to this subject of data integrity. So to begin with, what is data integrity? In the bottom line is data integrity is whether you can trust and believe the data, right? The data that's generated from your operations. Keep in mind that any decisions that are made by investigator, the agency, physicians, or any stakeholders are those decisions are based on the data that comes out of the trials. There have been major issues with respect to data integrity in drug manufacturing inspections and in new drug applications. And if you look, they rarely use data integrity, right? There are other terms that they use, which I'm going to go into in a minute. But unless it's really egregious, looking for the term data integrity isn't really going to get you much. Sometimes these are noted under the case history or documentation issues in clinical investigator inspections. I will say in the drug world, these have resulted in major product recalls, in new drug applications being denied because FDA didn't trust the data, and companies going out of business. So, when issues exist with the integrity of the data, basically all bets are off. Now, this should be a bright red flag. So what do these issues look like? Here are just a few examples found in 483s. So if you look, discrepancies between source documents and report forms. If you have two different sets of documents, two different documents, and they're saying different things, they don't agree with each other, then the question is, which do you believe, right? Which do you trust? What if there is no source documentation available? How can the data that's used for calculations or that's reported be trusted if you don't have the source document where it was originally documented available? Or if you have blank forms, there are issues with audit trails and changes that were made. And so these are just a few examples, of issues that were found. So let's look at what our model found with respect to these different types of data integrity in our 483 data set. And here's what we found, over half were issues with original data. So that's the original source data. Is it available? Is it reliable? Has it been altered, for instance? Also of issue is the attribution. So if it's supposed to be signed, was it signed and dated, or can you tell who signed and dated it, right? And then accuracy, which pretty much speaks for itself. So about 3/4 or so, over 3/4 really of data integrity issues in 43 observations, which again were found in over half of the 43s our model looked at, right, were due to original data attribution and accuracy. So here's some example data integrity language from 43 observations. The source documents indicated, right? But other documents indicated something else. Which do we believe? You don't have any documentation to support something. Again, how can we trust that? No information to identify the subject anywhere. Original source diary indicated something, but other documents said something different. One of my personal favorites, diary entry on day 6, is a carbon entry from day 5, which happens really all too often. So the question, if you're looking at this data from a CRO or an investigator or anyone, or if FDA is looking at this data, the question is, can you trust it? Right? Can FDA trust the data? Can FDA use this data to appropriately evaluate your studies, right? And do you have issues of this type in your operations? Something for you to give some thought to. So, I'd like to summarize. Basically, what I've shown in the last 20 minutes or so is that there are some rich data sets that can be analyzed. and they reveal some insights when we use the right tools to analyze them. And our models show what FDA is looking for and the deficiencies they're finding, but not at a high level of detail, but at an actionable level of detail. And the other thing I will mention is that these issues that we find and observations that we find, you can see them at a macro level, as we've talked about, but these can also each be mapped back to specific warning letters, to specific 483s. So that has a lot of utility as you think about due diligence. So I'd like to end up with telling you how Redica systems can help in various aspects of your operations. Study startup and ongoing monitoring. We have full regulatory histories on GLP and GCP vendors. Selection is sometimes not done in a very scientific or data-driven way, but we can help you do it in that way. Strategic inspection preparation. I've just talked about that for the last 20 minutes, looking for trends in the data and what are the things you really need to be looking for. Then tactical inspection preparation. And what I mean by that is we have complete FDA investigator profiles. So what do they look for? What do they cite? When this investigator comes in, we have all of these 483s. What do they tend to look at? What do they tend to find? So know what they look for when they show up at your door. Then we also have a regulatory and standards monitoring tool, which can keep you up to date, with the various GXPs and regulations and standards from the top 150 regulators and standards bodies around the world. And with that, I thank you. Here is contact information, both for my company and for me personally. So thank you for having me.
Okay, everyone, now we're going to go into our live Q&A section of the webinar. So please go ahead and enter. You can use the chat or the QA feature to enter questions. For starters, Jerry and Fran are going to touch upon some of the registration questions that you filled out on the registration form. So Jerry, I pass it to you.
Okay, great, thank you. So thank you everybody for coming today. When you signed up to attend today, you had the opportunity to submit some questions in advance. And so we looked through those questions, and a number of those questions, hopefully, I feel confident, were answered by the actual webinar presentation that you just looked at. And as far as some others go, I want to talk about a few general themes. We had about 30 questions come in, so I want to talk about a few general themes that address a number of those questions. Several people asked about data from outside the US. You can see that the model that we talked about today, that I talked about today, is really FDA centric. Well, the tools that we use have the ability to analyze data sets, not just from FDA, but the problem is getting those data sets, right? If you look at the EU, for instance, the General Data Protection Regulation, GPDR, which went into effect a few years ago, makes it really hard. There aren't any other agencies in the world that are nearly as transparent as FDA. And so our ability to get the FDA data sets is easier. Now, that's not to say that we can't do it. And we certainly are working on getting those data sets from the EU, and from other places. MHRA, as you know, publishes a lot of data on pharmacovigilance inspections. FDA doesn't do many, specific PV inspections, and so those are data sets that we're looking at. But right now, again, that's something that we're working toward, and we're very hopeful of getting those data sets and being able to analyze them. But we don't really have a lot of those now. Another major theme had to do with metrics on trends. Fran's going to talk a little bit about that, but I will mention that as you look at inspection types, as I said during the presentation, if we look at 483s enforcement documents, those 483s, about 75% of those are issued to clinical investigators. About another 12% are issued to IRBs. So that's 87% of that big data set. So that's where we chose initially to focus our efforts. Now, are there still sponsor, CRO, sponsor, investigator regulatory actions out there? Yes, there are. Are there pharmacovigilance inspections, REMS inspections? Yes, but there aren't as many of them, and so those are on our roadmap. But those will be coming later. So if we look at inspection types and specific inspections like remote inspections and trends in inspections, we've invited Fran, who works with us here at Redica Systems, to address some of those questions. So Fran, I'm going to turn it over to you.
Thank you, Jerry. Appreciate that. I just wanted to let you know that basically it's my background within the last 2 years, since I guess the end of 2019 to present day, I've gone through about 8 inspections, mostly BIMO inspections. And I think that 3 of those inspections were for PV, and 4 of those inspections were joint FDA, EMA, MHRA inspections. MHRA now inspects independently of EMA, so it's kind of a different, theory or different approach. Anyway, with these inspections, it was obviously during the COVID era, or I was at the end of the, I think 2 of my inspections occurred prior to COVID. But the difference between having inspections conducted during COVID versus not COVID was that the investigators and inspectors from EMA, MHRA spend a great deal more time looking through your documentation. They certainly want independent access of the ETMFs. FDA is more interested in requesting documents for you instead of basically having independent control of your ETMF. But the EMA, they will take control of your ETMF and look through documents, whatever they would like to do. And it's also very critical that they basically judge whether or not you have them trained on their system prior to letting them have access. And that includes the ETMF and the EDC. So it's important that you make sure that any investigator or inspector is trained prior to allowing them access to any of these electronic systems. I think that the main difference here is what I see is that there has been more observations and on a 483 nature, or just on a something that would be documented in an EIR regarding what they find in your documentation. So there are many more observations that they have regarding the requirements in the TMF plan, how your TMF is managed, what's contained in your TMF. There are many more observations now that are related to monitoring issues and sponsor oversight, investigator oversight. So I think those are the differences that I've seen recently with the COVID, during COVID, because they spend more time virtually reviewing documents and not in such a rushed situation where they come into your site and they're only there for a week. In fact, MHRA, I think their average amount of time that they spend in the inspection process is over 4 weeks. So.
Thank you for that insight, Fran. We do have a couple questions if y'all would like to move on to those.
Okay.
Sure.
Okay. Are remote data reviews by the inspectors here to stay?
From what the communications that I'm getting from the last 8 inspections that I got is the answer to that would be yes. They would like to be able to have more time to review documents, to upload them. In fact, it's interesting because FDA, at least, they want you to upload into SharePoint. They'll have you upload into Box, and they don't really ask a whole lot about how secure it is. They just want to have access to those documents. So I think that, yes, remote review of your documentation, your study documentation, is here to stay.
Okay.
I would add from kind of looking from the GMP side and just looking in general, what's happened over the past couple of years with the pandemic has been that so many things have changed, right, around the world, and so many places. I mean, everyone is kind of looking at their staffing models, looking at their work models. You know, is this the right number of people? Do people actually have to come into the office to do their work? You know, or can they do it remotely? And so those kind of assessments are going on a lot of places, right? And government agencies are really no different. I will mention again, and this is on the GMP side, but I know we have people who kind of cross into both of those areas. On the GMP side, FDA is doing remotely what they're calling assessments, right? They do not call them an inspection. For it to be an inspection, a Form 482 needs to be issued, notice of intent to inspect. And so they're doing what they're calling assessments. So that's, you know, that's a little different. But the FDA investigators and FDA personnel I've talked to have said, you know, getting a lot of these documents in advance and doing these document reviews before we ever get on site, we have learned by doing that that can be a really helpful approach. So a lot of words to say, yeah, I agree with what Fran's saying. It is certainly here to stay, at least to some extent.
Okay, thank you both for those insights. Another question we have is, what are expectations for handling routine temperature excursions, frequently encountered drug shipments of drug products to clinical sites?
So, Jerry, do you want to answer that? Because it's, you know, both of us, it's sort of in the GCP world, it kind of, we adopt basically what occurs with GMP, that this is a very, very common problem. And especially with in-use stability temperature excursions, where the IP has been already prepared for dosing, but it sits around, or they transfer it from one site to a couple blocks away, so without temperature monitoring. So it's a common problem. But I think that there's a number of things that you need to make sure that are implemented before you ship any IMP for your clinical trials, and that is that you qualify, validate your shipping process. And I think that FDA definitely does come in and want to review that type of information. So Jerry, you could go ahead and elaborate on what's required from a GMP standpoint with that.
Sure. Thank you, Fran. Yeah. Really, when you're talking about a temperature excursion, you're talking about product quality, the quality of the product, whether it's an investigational medicinal product or whether it's a finished drug product that's in transit to a pharmacy somewhere. It's really the same thing. The agency expects you to have systems in place to prove that the temperature excursion, if it's a temperature sensitive product, that that the temperature has been properly maintained, and that requires validation, qualification of shipping, of shippers, the use of indicators. For example, there are some called temp tails that actually accompany the shipment. So if you have a product and it is going from one place to another, and it sets out on the tarmac of an airport because the jet is having mechanical problems, and it sits there for several hours, then the question will be, did it maintain proper temperature? Is this material still good to use, or is it not? And can you prove it? right? So yeah, there have been major recalls. There was one recall that happened, I believe, in 2017 at a quite a large company that had to recall, I believe, it was 112 products because they were all in a warehouse that lost temperature control, and they did not have the documentation to show that once those products that were all supposed to be refrigerated warmed up above the appropriate temperature, that they maintain their quality. So it is a big deal whether it's a final drug product or it's going into a clinical study.
Right, and I think that I'd like to add another couple of other points, and that is when you're conducting a clinical trial, from what my experience has been with these last inspections, is that the regulatory agencies want to know and be able to see documentation of training that the pharmacists or the study coordinator who's ever involved with or been delegated the responsibility for the IMP, that they're trained on how to use the temp tails, trained on the stability, temperature, allowable temperature ranges, and that training is documented. And also what I've had occur recently is even the FDA reviewers of the IND have requested the pharmacy manuals, including phase 1 clinical trials, to take a look at how that whole process is taking place and how it's described in the pharmacy manual, and where the pharmacists at these clinical study sites, do they have a contact number to call the sponsor if temperature, if the temperatures are out of range. So it's very important to have all that documentation in place for temperature monitoring, because FDA is really catching on to all of this and now really taking a closer look at how all that's managed.
Yeah, just one final kind of footnote, and that is the bottom line. A lot of times we talk about regulations and what are the regulations? What's the letter of the law? What's FDA look for? What are you going to get cited for? Really, the bottom line has to do with the patient, right? The drug that's being given to the patient, has it been compromised? Is it still safe? Is it still effective? And there are a lot of ways that it can be compromised, and we need to be sure that we can show that it wasn't before it reaches the patient.
Agreed. Thank you both. Next question we have is, how do you link the source data category to the reduced SDV % due to RBM, for example? Should it be a signal to increase SDV?
From a remote aspect? Yes.
Yes.
My answer to that is yes. You need to make sure that if this is a monitoring process, which is different than an auditing process where you go in and take, you have an algorithm for basically figuring out how many documents that you want to review, or study documents that you want to cross-reference with the source document. But from a monitoring aspect, that's more of a quality control process, and you should be doing 100% source document verification for all critical data points. And Jerry gave you a pretty good description of data integrity, and data integrity has to do with your critical endpoints. So you need to make sure that you are doing 100% source document verification of all of your critical endpoint data.
Anything to add, Jerry?
No, it's a pretty complete answer. Thanks, Fran.
Perfect. All right, next question we have. What is meant by inclusion and exclusion criteria in the context of a 483 citation? Does it mean that the study included patients that didn't match the inclusion criteria, or patients that should have been excluded but weren't?
Fran, I can take this just at a high level based on what we build into the model, and you can get some detail if you'd like. But according to to the model that we built, what we found in 483s was there were a lot of issues with both inclusion and exclusion criteria. And what that means is if you have a patient who is participating in a particular clinical trial, there are certain kinds of conditions and attributes of that patient that you want included in that clinical trial or that you want excluded. So for example, if it says that a person, that this trial is for only for women who are not pregnant, and you include a pregnant woman, right? Then that's an exclusion criteria, right? That's been violated. So it's really all around as you're testing a product, testing a therapy. Who is the intended patient population, and what patient population is going to give you the kind of results that you're looking for in the trial that you're running. So people need to be included and they need to be excluded for certain reasons, and that doesn't always happen.
I think that from my experience as 40 years of auditing clinical studies, that the biggest problem with inclusion exclusion criteria, and also I think that FDA and the EMA find this to be a common problem with them as well. And that is that when patients are included and you have these inclusion criteria and exclusion criteria, what happens is that there's no objective evidence to show that that patient was actually qualified for the study. There may be one particular point that is missing. It's part of that inclusion criteria. And if you can't have objective evidence, if you find no objective evidence to show that that one particular inclusion criteria was not met, then that patient should not be enrolled. Also, there's a lot of talk and observations around the fact that, hey, a patient doesn't make it through the inclusion exclusion criteria the first time, so they bring the patient back, months, weeks later, after they've been allowed to do certain things, and then they include them in the study because they are able to pass the inclusion criteria. However, there still isn't a good objective evidence for that. So I think it's very important that, and what the main problem I think with inclusion exclusion criteria is the lack of objective evidence, and the fact that some companies still allow their clinical study sites, their investigators, to provide protocol waivers to get them into the inclusion of the study. And it's, MHRA and FDA do no longer accept waivers. So you need to be very careful with that when dealing with inclusion exclusion criteria.
Thank you both for that insight. Good to know. This next question goes back to one we touched on previously for temperature excursions. So for products that require refrigeration and then have a temperature excursion, is time out of refrigeration and supported data for the time out of refrigeration based on validation or accelerated stability studies acceptable to the FDA?
This really depends on what you're talking about. If you're just talking about the IP in a vial, your stability studies should include that. And I think that Jerry could probably talk about that. But the in-use stability is also very important. And that is that if you have constituted the IMP, such as in an IV bag or whatever, which is very, very common with biologic study, immunotherapy studies, and that sits around in the room for more than 6 hours, FDA expects you to have, in fact, that's the key issue, 6 hours. If you go over 6 hours, you need to have in-use stability data to show what happens to that IMP when it's outside of the refrigerated area. So I think that it really depends on what you're talking about. If you're talking about just the drug prior to in-use preparation versus post in-use preparation. But both times it does require objective evidence to show that the product does not degrade once it's been kept out of the refrigeration for a certain period of time.
Mm-hmm.
The in-use aspect is something that's very specific, here to clinical studies. It doesn't come up on the GMP side at all. Now, on the GMP side, it really has to do with If you had this under refrigeration and took it out and you were going to use it and it was in a vial and it set out for an hour and you didn't use it and you put it back, is it okay? As long as you have studies that mimic what has just occurred with that product and shows that it's okay, then you're okay, right? There aren't any exact specific written down requirements. You have to do this and this and this and this, right? You just have to be able to show that whatever occurred with that product, that you have bracketed that in some kind of study and tried that, and the product was fine. So that's the kind of stability data that they're looking for.
Right. And I think that FDA now, remember that FDA, they may not have regulations that are specific because they're always lagging behind before they come up with a guidance document or whatever, but they fall back to industry standard. And industry standard is that if you go over 6 hours, you need to have chemical and microbial challenge studies. So it's very important that, that's done, that you perform those studies prior to going over a 6-hour in use outside of the allowable time frame.
Okay, thank you. Last question that I have here is CAPA and design still a hot area that inspectors like to see?
Yeah, I'll start with that. I mean, CAPA is always a hot topic. It always has been. I think it always will be. It's a corrective action, preventive action. In other words, I mean, it goes to the heart of good practice. It's whether it's good manufacturing, good clinical, good laboratory, whatever good practice is. In other words, you have a particular sequence of events. You have something that's supposed to happen, something that's supposed to take place, and it doesn't. Something different happens. There is an outside event that happens. There is a mistake that happens. Something happens that's different, and you have to be able to detect it, and you have to be able to show that it didn't have an issue. It didn't cause an issue. That there's no impact, or if it has an impact, that the impact's acceptable. And then from a preventive action standpoint, you have to say, well, we had this problem and we corrected it, and now we've put a system in place so that it won't happen again. Right? I mean, that's really, in a lot of ways, that's just common sense for any kind of process. And I think that always has been and always will be something that investigators will look for and that companies need to build into their systems. What do you think, Fran?
Well, I think that you have to be very careful with in clinical trials. I've had experiences. Remember that when FDA comes in to inspect you for a BIMO inspection or PAI, they always ask for your CAPA log, your CAPA list. And, my experience has been that there's a lot of like CROs who conduct audits and whatnot, and every observation is a CAPA 1, CAPA 2, and that's just not what the proper use of a CAPA. CAPAs usually occur when you have critical or major observations or critical or major quality events. FDA now is really tying CAPAs from clinical trials that I'm looking at into risk management. What has been your risk management procedures? How do they relate to you issuing CAPAs? How are you trending these major critical issues that can't be reconciled outside of the CAPA realm? And remember that CAPAs require, you don't issue a CAPA until you do an investigation. You do a root cause investigation. So it's very important that you don't just issue a CAPA, that you have defined what the root cause is by an investigation prior to issuing a CAPA number.
That's a really important point. I mean, I tend to think of CAPAs in the context, because of my background in GMP manufacturing, of being in a manufacturing facility and something happens. But I have seen exactly what you're talking about, where an investigator comes in, there's an inspection, and there is a laundry list of items, and a company assigns a CAPA to each one. And I 100% agree, that is not the correct use of CAPAs. There are some things that can just be fixed, right? And if you don't have a root cause, that you're trying to fix, then instituting a CAPA is just a waste of time and resource, and it's probably not going to help you anyway.
Right, and you need to make sure to train your monitors about the proper use of CAPAs. In their monitoring trip reports, they should not be saying, you know, we should issue a CAPA. That's something that needs to be adjudicated, and you need to kind of talk about that before you go ahead and issue a CAPA. So that's very important. And remember that, you know, when FDA comes in, they look at your CAPA lists and they try to determine how long it's taken you to resolve that CAPA. And have you trended to see if there's any trends where you may need to go amend your protocol or, you know, have a backstory on the CAPA. So it's very important that you really make sure that you're using the CAPA process properly.
Thank you both. We actually had another question come in, so that was not our final question. So last one we have is, has FDA started back up for foreign clinical investigator GCP inspections since COVID?
From my experience, they're trying to do that, but I don't think that, I think there's still a pretty big backup, and especially I know that they've really now kind of cracked down on some of the studies that are conducted in China. If you've looked at some of the publications that they've had recently, and I don't know, Jerry, if you're aware of this or not, but FDA now will only accept less than 20% of data that comes out of some of the clinical trials that are conducted in China. And one of the reasons for that is that they're really backlogged on doing inspections there. So I think that, yeah, there is still a backlog.
Anything to add, Jerry?
No, I mean, it's pretty much the same thing on the GMP side. We're still revving up, you know, they're still revving up their systems for overseas inspections, and it'll take a little while for them to ramp back up. And regarding Fran's comment about China, you know, we do see a lot of issues there on the GMP side as well, and so that's an area to be aware of.
Okay, well, that concludes today's webinar. Thank you, Fran and Jerry, for joining us and answering all these questions. If anybody in the audience still has questions for either of them, you can email back, and we will be sending out a recording of today's presentation, as well as to any questions we have not gotten to. Thank you all for joining, and we'll see you at the next webinar.